And we have Dr. William Telford from the NCI, who is here today. And he's one of the top experts in flow cytometry. We've known each other for a long time. He came here about 2 years ago. He gave a nice talk on what flow cytometry can do in its basics. And so I thought it'd be nice to have, since he was coming to Ahmedabad again, I thought it would be nice to have a follow-up conversation about what are the advances in this technology that can help us kind of answer questions in biology that other techniques may not be able to do so well. A brief background about him. The bio is there in the email, but I'll tell you my own version of his bio, right? So as I said, we've known each other for what, more than 20 years now. And he's been teaching and we've taught together in many of the workshops. There used to be a technician in his lab, Veena, and one of the complaints that Veena made to me was that in our lab, no machine is ever closed. Everything is open. And so it's so nice to see a box that just works. So that's something which never happens in Bill's lab. Okay, every machine that he gets is opened up. He's done a lot of work, even worked with manufacturers, trying to standardize some of the newer instrumentation, develop applications for those, or even develop standardized reagent applications. So I thought it'd be good we couldn't find a better person to talk about where this technology is headed and essentially what it's going to do for us in the coming future. Yeah. Welcome, Bill, to Ahmedabad.
So how many of you have done flow cytometry yet or know something about it based on the equipment that you have here? Because you have some nice equipment here. So I'll talk about some of the basics, but I also want to show you how far we can go with it. Because when I first did flow, it's a very powerful technique, but when I started it, the capabilities were I'm old enough that I remember when the capabilities were very limited. Now there are instruments that can do up to 50 cellular markers simultaneously. We're big data now. Took us a while, but we're big data. And I'll focus particularly on something called spectral cytometry, which is a newer technology that is really making this possible. So who am I? I run a research core lab at the NCI. This is our building in Bethesda, Maryland here. I'm not a professor. I'm a PhD, but I run this core laboratory. We support both basic and clinical research. And as Vivek says, we like to open up instruments, see what they can do, and try to improve them. This is something that's a big part of my laboratory. So for those of you who are not that familiar with flow cytometry, this is how a cytometer works. First, we need a biological sample. We label it very often with one or more, sometimes a lot more markers. We run the cells through a light source, usually a laser. We detect the light scattering and also the fluorescence from those cells. We then use optical filters and mirrors to separate out the different fluorescent signals. And then we take that analog data, convert it to digital, and we analyze it. And one of the big things we do with flow, not the only thing, but one of the big things is look at peripheral blood, looking at all the different types of subsets of cells that are within a peripheral blood or in another immune tissue. And if any of you have done differential counts, if you've worked in medical technology, you can simply look at the light scattering from a laser being shined on a cell to measure both the size and also the optical density of the cell simply based on light scattering. Hematology analyzers work this way. But you can then take that technique further and actually identify what the cells are by using antibodies that are conjugated to fluorescent probes. And there are hundreds of these out there. These fluorescent markers, like all fluorescent molecules, have an excitation wavelength and an emission wavelength. Fluorescein is probably the one you're most familiar with, and what makes Mountain Dew green. It excites in the blue range of the spectrum and emits out here in the green. It's a very common biological stain that we use for microscopy and a lot of other techniques. There are many others as well. Phycoerythrin is an orange emitting dye. We can attach these to antibodies, and then we can add these antibodies to our cells and label them. And this is what the data ends up looking like. First of all, as we did with scatter, we use a laser to excite those dyes. And then we use filters and mirrors to collect the fluorescent signals from those dyes. If a cell has a lot of a marker, it'll have a strong signal. If it doesn't have so much, it'll have a weaker signal. And then we can express that data in either 1 or 2 parameter plots over here. So this is a histogram, for example, for a marker CD8 that's on cytotoxic T cells you may know about. CD95 is a marker associated both with apoptosis and also cell growth. But what makes flow cytometry really exciting, in my opinion, is that you can look at more than one marker simultaneously. You don't have to look at just one of these at once. And you can express that data in these 2 parameter plots where each of these pixels represents a cell. And we can see, for example, that we have CD95 expression in both our CD8 negative and our CD8 positive cells. So flow cytometry data is correlated and interrelational. We call it multi-parametric because you can look at more than one characteristic at a time, which is immensely powerful. We can look at very complex mixtures of cells. This is only a 2-color labeling. We can go up to 40 or 50 at this point, and I'll tell you a little bit about that. Now, back when I was young, many, many years ago, I was an undergraduate at the University of Rochester in New York State in the US. And I was lucky enough to work, to volunteer at the cancer center there in the laboratory of Leon Wheelis. Leon was one of the pioneers of flow cytometry. He died very recently. But this was the machine he had. This is an early flow cytometry. It's huge. It's got a big computer on it. It's got a big laser over here. It could only look at 1 or 2 colors at a time. But at the time, this was the state of the art. So it had a single laser source and could look at fluorescein, prapidium iodide, lots of other dyes. This is the system that I used as a graduate student. I was in grad school back in 1989. And this machine was already old then. It's called a cytofluorograph. It also had only 2 fluorescence detectors, but it had lots of laser wavelengths. It had these big water-cooled argon and krypton ion lasers that not only could produce the blue laser that excited fluorescein and other dyes, but red and yellow and violet and ultraviolet. So we were able to excite a lot more dyes that could measure different things in cells. And then a little bit more recently, this is came out in 1992, was the FACS caliper. You may see this instrument in some labs you work in. There are still FACS calipers being used today. Huh? You've got one here. Absolutely. So now we're starting to move up in detectors. We can look at 4 different fluorescent markers simultaneously, plus those scatter measurements for cell size and optical density as well. This is an instrument we have at the University of Cape Coast in Ghana, still in use, still being used all the time. And if you open up the cover, as Dr. Vivek said, I love doing this. You can see everything inside the machine. The lasers are here. There's a blue and a red laser. The detectors are inside this box over here. By looking at more fluorescent markers and more markers on cells, we can get more information. This is what the optics look like inside the machine. We use filters and mirrors to take all these different fluorescent signals and split them into green or yellow or orange or red. We're able to separate the fluorescence from these different markers and look at what is attached to these fluorescent probes. And this is what the spectra look like over here. What was really exciting about this machine when it came out was there were 2 laser wavelengths here. So we could look at 2, the markers that were expressed or excited by 2 laser wavelengths. And this is the type of data we collected. Rather than just looking at 2 markers, we're now looking at CD4, which is on helper T cells, CD8, which is cytotoxic T cells, CD45, which is on all leukocytes, and CD19, which is a B cell marker. And each of these little clusters here, each of these pixels in these clusters represents a single cell. We can see the CD45 positive cells, which is most of the cells in the peripheral blood, which is what we're analyzing here. We can see the B cells and also all the T cells, but also the CD4 and the CD8 positive T cells. This is a 4-color panel looking at 4 important markers in the immune system. And this panel is still used to this day to do CD4 counts in patients that have HIV. So by adding those extra lasers and those extra detectors, we're getting more information. And it's simply grown from there. This is a flow cytometer made by a company called Agilent, ASEA Biosciences. It has 3 laser wavelengths, a blue, a red, and a violet, and 13 fluorescent detectors. So we're now getting a lot more information. And if you look at the different dyes we can use that can be attached to antibodies, we are able to do 5 probes, 5 markers off the blue laser, 3 off the red, and 5 or 6 off the violet. We've added an additional laser wavelength and are going to get more information as a result. Oops. Sorry, let's back up. And as you can see, the data is getting more complicated, but it's also becoming more informative. To that 4-panel color panel I showed you before, we've added some additional markers here. CD45RO and RA are T cell markers that are expressed on naive and memory T cells. We've added a natural killer marker, CD56, and we're able to go in and identify all these subsets. The data is getting more complicated. It's still not so complicated that we can't do it manually, but we're getting more information and more precision by adding in new laser wavelengths and additional markers. Now, this can start to get kind of crazy. This is a BD Biosciences Symphony A5 flow cytometer. This instrument has 5 lasers and 28 fluorescent detectors. So we can do what we are now terming precision medicine or precision analysis, where we can really burrow in and look at the fine structure of the immune system. This is the inside of the instrument. Again, we love taking the covers off things. This is the flow cell that the cells actually pass through, and you can see the different lasers shining on the flow cell where they're going to excite the cell and ultimately have the fluorescent signals transmitted back to detectors inside the instrument. Yeah, sure. Right. We're going to use more laser wavelengths, and that will allow us to use more dyes, and we can add more markers to our panel. So the same principles apply, but instead of just using 1 or 2 lasers, we're now using 4 or 5, and we're using fluorescent probes that are excited by those lasers. So these are the 5 different laser wavelengths, and we have fluorescent tags that can be excited by a green laser or a red laser or an ultraviolet or a violet laser. So the same exact principles apply, just adding complexity. We're adding more markers to our panel. Does that make sense? Okay. So the instrument is getting pretty complicated. Yeah. It can add to that, and it can also, because you have to separate out all these colors from each other, and I'll talk about that, doing something called compensation. It does, and it becomes more complicated. And once you start looking at this many markers, you have to very carefully plan what fluorescent probes you're going to be using and what markers what antibodies they're going to be attached to for what markers. We call this panel design, and it becomes very important because, as you say, some of these fluorescent tags may spectrally overlap with one another, and you have to separate them out. We have all these filters in here that will begin the job, but it's not the end of the job. And I'll get into that because that's an excellent point. Things are getting more complicated. The nice thing about flow cytometry is that the cells only spend about a microsecond in the beam. So photo bleaching isn't a huge problem with flow like it is for microscopy. But there are other phenomena that cause that as well. And we can talk a little bit about that too. It means we have to be careful about the power we use on our lasers. Fortunately, cytometry is very fast. The cells are moving at about 10 meters per second. And we can analyze thousands and thousands of cells in a very short time. But that can be an issue. Absolutely. So this is what the data looks like. This is this is in a journal called Cytometry, which is the flagship journal of a society that Dr. Vivek and I both belong to. And they have a publishing format where people can publish very well-designed panels that can then be used by other investigators. This is a 28 color panel. So we started with only 4, now we're up to 28. And you can see the data is becoming very complex. We're able to look at T cells, B cells, dendritic cells, NK cells, all sorts of things. But as you say, we're now getting to the point where we really have to design our panels very carefully. And the data is becoming big enough that it's very difficult for us to simply look at it and draw conclusions. We're starting to, you have to use the same probability state analysis tools that the genomics and the proteomics people have been using for a long time. We're starting to have to use those for our data analysis as well. Absolutely. For example, sometimes we distinguish cells not just by whether they have a marker, but whether the marker is dim or bright when it's expressed. Absolutely. And although I'm talking about cell surface markers and protein targets in cells, we have markers for cell physiology, how well the cell is proliferating, if it's undergoing apoptosis. We have many fluorescent tools. We won't be able to talk about all of them today, but we have a large toolbox of fluorescent probes that we can use in flow cytometry to look at many aspects of cell physiology, not just the markers that are expressed on the surface over here. But we're really starting to increase our sophistication. And this is really the upper limit for conventional technology. This is a 31 color panel. These are all the markers that are being looked at. So we're looking at things like T cell and B cell markers, but we're also looking at transcription factors, cytokines and chemokines, cytokine and chemokine receptors, intracellular proteins, nuclear proteins, things like FOXP3 as a transcription factor. We can look at many, many things using an antibody tagged to a fluorescent probe. However, although this is very sophisticated, we are relying still on what we call the one laser, one detector principle, where we have a laser wavelength, it excites a fluorescent probe, and we detect the fluorescence. We may have many lasers and many fluorescent probes, but this is still what we call conventional cytometry, where it's one laser and one detector. And right now, 31 color is about the upper limit of how far we can go using the conventional technology. Now, 31 is a lot. And you might ask, do I really need to go higher than this? We're reaching the point now, though, where we are aware of thousands of different receptors on cells. And even within a single cell type, like T cells or B cells, we might know about dozens or even hundreds of different markers that we might want to look at. Remember that cytometry data is multi-parametric. We can look at one marker within the context of all the other markers. We're not looking at them separately. So being able to achieve this level of panel design is actually very important if we want to do precision analysis of the immune system. We're still not at the point where the genomics, people in the genomics world can look at thousands of things at once, proteomics as well. We're probably not going to get to that point, but we're definitely moving into the realm of what we would call big data. And we're going to use the same tools that they use. So you brought up the idea that we have to be able to distinguish these markers from one another. And as you start moving to larger numbers of markers, that can become difficult. One of the tools we use to distinguish these different fluorescent probes from one another is called compensation. Because, for example, in a very simple example, fluorescein and PE over here, FITC and PE, our green and our orange marker, you can see that they overlap with one another. And if you label some cells in this dot plot over here with a green marker and an orange marker, a FITC and a PE marker, you can see the FITC fluorescence is showing up a little bit, quite a bit actually, in the PE detector. And if you were able only to look at this in one parameter, this might actually look like PE, like CD8 PE expression over here. We use a tool called compensation where we're able to electronically subtract out that overlap. So it's a mathematical calculation. Basically, we use mathematical tools to subtract out that overlap and have our fluorescein, our green marker expression over here, become equal to the unlabeled in the PE channel and vice versa. So we end up with something called compensation values over here. The amount of compensation required to get the green signal out of the orange is pretty high, about 28%. We express it in percentages. Not so much for the PE into the fluorescein over here, where it requires just a little bit of compensation. But this is something that a tool that we have to apply to conventional flow cytometry data. Now, this is what we call a compensation matrix. It is all these values over here. Here's a 2-color example. This is a 4-color example. And you can see we already have 14 different values that we have to apply to that 4-color data that I showed you initially that came off of the old Excalibur. Once you start moving into higher dimensional data, it's hundreds of values. And this is only a 13-color panel here. And you can see some of these required subtractions are quite high, so high that they can start to compromise the integrity of the data. There are other tools that we have for looking at this as well. But for conventional cytometry, compensation or spillover subtraction, as it's sometimes called, is important. And achieving reasonable values and also something called data spreading, which I won't talk much about today, becomes much more difficult as you add more and more colors. There's also something called data spreading, where when you have to do that subtraction, it can spread out the distribution of the data and add a lot of error into your analysis. This is something that's become a very important issue in high dimensional, as we call it, flow, because we have to introduce a lot of these compensation subtractions to separate out the different markers that we're looking at. So it has a very big impact on the overall data. And although the details aren't very important today, what this means is that when we design our panels to look at these things, we have to take a lot of care to make sure that we use the right fluorescent probes that are not going to overlap into each other significantly. So that is conventional cytometry, the one laser, one detector approach. But we now have a new tool available to us called spectral cytometry. Conventional flow cytometries, as I said, use single lasers to excite a fluorochrome, and then we detect green, orange, yellow, red fluorescence. Spectral cytometers collect all of the fluorescence data simultaneously. So while if I was running fluorescein, I would just detect a single green range of fluorescence here. Spectral cytometers will detect all of the fluorescence using all of the lasers. You're taking everything. If any of you are doing multicolor microscopy now, you may already be familiar with this concept. The microscope people have been doing this for quite some time. Now, collecting whole spectra rather than just narrow bandwidths has a number of advantages. We can now use what is called deconvolution or unmixing, spectral unmixing, to separate our different fluorescent probes rather than compensation. This has more integrity. We have a lot more granularity in our data, and I'll show you how that works in a few minutes. So this is what a typical spectrum looks like. I have a cytometer, a spectral cytometer that has 5 different laser wavelengths, a UV, a violet, a blue, a yellow, and a red. And each of these little tick marks down here represents a single channel moving from shorter to longer wavelengths, 16 on the ultraviolet, 16 on the violet, 14 on the blue, and so forth. We end up with what we call a spectral signature, and we can use those spectral signatures to separate out our data, our different markers, and get the same data that we're getting from conventional cytometry. So this is how we do it. And there are now a lot of spectral cytometers on the market. And if any of you have taken physics and have done an exercise where you're separating white light into its various component colors, that is what's all that spectral cytometers are doing. They're taking all those different fluorescence emissions and separating them out into their components. So Sony Biotechnology, for example, they came out with the first spectral cytometer. They use prisms, just like Isaac Newton did. You take all that mixed light, separate it into different wavelengths, and then pick up the different markers using an array of prisms. Their newer machines now use diffraction gratings, which are just glass surfaces that have millions of little prisms on them, a little more efficient over here. The instrument I have in my lab is called the Aurora. It uses an array of solid state detectors with mirrors here that take that colored light, bounce it down a pathway, and separate out the different colors from red all the way down to the ultraviolet. And there are other systems as well. BD has a system. Agilent now has a system as well. This is becoming common. And there will reach a point someday where all cytometers are going to do this. We're not at that point yet. Conventional instruments are still very, very useful, and you've got a bunch of them here. I still have a bunch of them in my laboratory. They're not going away. But I do want people to be aware of what the spectral cytometry technology is, because this is a new thing in flow, and it's going to become more and more dominant. So the instrument that we have is called an Aurora. It's made by a company called Cytec. It has 5 laser sources, and then it has these arrays of detectors that pick up full spectra, 64 channels total. We take all those spectra. For example, in this case, I'm analyzing, I'm not sure what I'm analyzing, probably Lexaflor 488, which is a green dye. We take all those individual spectra, we merge them together, and then we get what we call a spectral signature, like I told you about earlier. Now, if we're going to be looking at more than one marker, and we are doing this in cytometry, you collect a spectral signature from each of the dyes that you are using. These are all fluorescent probes, violet, a green or a yellow, and a red excited probe. And you can see the spectral signatures are a little bit different from one another. The more different they are, the better. However, they don't have to be extremely different from one another. And this is why spectral cytometry is a better technique in some ways than conventional. We have much more information to separate out those fluorescent tags in our panels. So rather than doing compensation, like I showed you earlier, we take our spectral signatures and we do deconvolution to separate out the fluorescein from the PE and the PE from the fluorescein. So I showed you that there was some mathematical, some somewhat high mathematical value here to separate PITC and PE. We're able to do it very, very easily with very little mathematical manipulation because these two dyes are not very similar to one another. This little number is called a similarity index. It ranges from 0 to 1, with 1 being identical and 0 being completely dissimilar. And the system really does not consider PITC and PE or Alexaflor 488 and PE to be very similar to one another. So we're able to separate them very, very well. And if we're doing more markers, we get what is called an index, a similarity index matrix, where for each of the fluorescent tags we use, we get a value ranging from 0 to 1. 0 is no similarity, and 1 is identical. And we have some combinations in here where there's a lot of similarity between the two dyes that we are using. For example, Pacific Blue and BV421 are both violet excited dyes. They emit in the blue. They're very close to one another spectrally, but they are far enough apart that we can still use them together. We could not use these two dyes by conventional flow. And here's an example. We have 2 different markers, allophycocyanin, which is a natural product. It's a red excited dye, emits in the long red, and another dye called Alexaflor 647 that is a synthetic dye. It's low molecular weight dye. But if you put these on a spectrophlorimeter, you can see the dyes are very similar to one another spectrally. And by conventional flow, we cannot distinguish these together. We can't use them together. However, If you look at the spectra, not just with the red laser where they're excited best, but all the other wavelengths, the signatures do differ from one another using some of the laser wavelengths that we're exciting the cells with. And we're able to separate out the signals from these two dyes. That similarity index is a little on the high side. It's getting up above 0.8, but it is still sufficiently low that we can use these two tags together. What this allows us to do is design bigger panels and look at more markers, because we can now combine these two markers together by spectral flow where we couldn't do it by conventional flow. So we can now attach these to antibodies, look at two different markers, and separate out those signals. And I'll show you a very good example that we did in my laboratory a few years ago, showing just how powerful spectral cytometry is. There is a series of fluorescent proteins out there, like GFP, that emit in the up in the infrared, the near infrared. They're called the IRFP proteins. They're bacterially derived, and there are 5 of them, ranging from IRFP 670 up to 720. And I can set up one of my cytometers with a couple of different red and long red laser sources to separate out these dyes, to allow me to use them simultaneously. There are circumstances where we want to combine multiple dyes together. But it doesn't work that well by conventional flow. You can see just how close the emissions are. These are the excitation and emission spectra over here in the upper right-hand corner. And they're very, very close to one another. We really cannot separate them by conventional cytometry. However, if we use a spectral cytometer, and in this case, we're using a Sony instrument, these are the spectral signatures for the 5 different dyes, and we're able to separate them very well from each other. There's just enough difference between them that we can pull them apart. We can now use these 5 dyes together. We can build big panels looking at a lot of different markers simultaneously. So here is a pretty big panel. This is a 23 color panel. It was designed for looking at human T cells in graft versus host patients by a laboratory at my institute, the Canacry lab. Here are the different fluorescent tags in the left column. In the right column are the targets, are the proteins that the antibodies are going to be binding to. Don't worry too much about all these names. There are hundreds of different fluorescent probes out there that have all sorts of different excitation and emission conditions. But this is what the data looks like over here, where we're looking at scatter, and then we're able to separate the cells into T cells, B cells, NK cells, dendritic cells, and look at things like their transcription factors and other markers as well. And this is what it looks like by spectral. That little similarity index I was telling you about, where each of these little squares represents the overlap of one fluorescent tag to another. And although we have a few hot spots in here, this is a pretty good panel. Everything looks pretty well separated. And even some of these dye combinations here, where the dyes have very similar spectral characteristics, are just dissimilar enough that we're able to pull them apart, something we could not do by conventional cytometry. Yeah. Yep. It's a function of only the spectrum of the dye. And while those compensation numbers are actually what's the information that provides separation, this is more like a chi-square, where you're looking at whether the dyes are similar to one another or not. This is not what actually does the separation. It's to let us see how much overlap there is. Back when spectral got started, we didn't have this. And it was very upsetting to us, because most of us are used to seeing these compensation matrices where we could say, okay, 2 dyes are working well together, or 2 dyes are working badly together. And we couldn't do that. So they generated this little algorithm here that would let us see whether one dye was overlapping with another. But this is a good indicator of whether we have a compatible group of markers. Are we going to really be able to tell them apart? And we have dyes here that are very close to each other spectrally that we are able to separate into distinct populations. Nope, nope. When I say overlap, I'm referring to the fluorescent emission. So if you have a green dye and a yellow dye, their spectra are going to overlap with each other a little bit. You know, we have filters and mirrors that are separating out the signals, but they're not perfect. So if I just jump back real quick to here, to those two red markers, APC and Alexaflor 647, these are the emissions, and you can see they're almost the same. So it would be normally very difficult to distinguish one of these dyes from the other, to pull those signals apart and use both of these markers simultaneously. So when I talk about overlap, I'm talking about the fluorescence emission. And however, to your point though, a lot of the markers we're using, some of them are on exclusive subsets like CD4 and CD8. Some are not. Some are expressed on the same cell. And choosing dyes that you can separate from another, making that right choice becomes very important when that happens. Because if a cell you have has both of these antibodies with both of these dyes, you have to really make sure you can separate out those signals or you can get erroneous data. Yeah. Uh-huh. Mm-hmm. Okay. Yep. Mm-hmm. Well, yep. This is not a combination we would normally use unless we really needed to use a lot of markers. We do flow cytometry on plants. Absolutely. Absolutely. No, we do it on plants. We don't have so many protein targets in plants, but for example, we'll measure the DNA content in plant cells, can certainly be done. So flow cytometry is done on plant cells that have been dissociated from one another. We can talk about that because that is, and the machines you have here can do it. Absolutely, they can do it quite well. So I'm just showing this example just to give you an idea of if we needed to build really big panels and we had to use dyes that were pretty close to one another spectrally, we could combine them together. So let me jump ahead here. Okay. So this data I just showed you, the this 23 color panel, because we are now moving into the realm of big data, these 2 parameter plots here are very limiting. It can be very difficult to interpret what's really going on in these populations because it can be hard to look at one marker and say, okay, what's happening with the other 22? Our brains just aren't well equipped for that. So we're now starting to rely on the same probability state analysis techniques that the genomics and proteomics people have been using for a while. We're starting to rely on them as well. So for that 23 color panel, we can look at these, they're called Tisney-Visney plots. Each of these clusters represents a distinct cell type, and we can go in and look at the relationships between these markers and all the other markers that are present in our samples. And this is now becoming very common in what we call high dimensional cytometry, where you're looking at 15 or 20 or 30 or 40 or more markers. So we're starting to rely on these tools much more and more. So how high can you go in spectral? This is a 40 color panel that was designed by my very good friend, Maria Hymas, who works at SciTech. And although the print is very small here, what you're seeing here is a large collection of leukocyte markers, T cells, B cells, dendritic cells, NK cells, every monocytes, macrophages, transcription factors, everything. And this is what the data looks like over here, where we're looking at each of these little boxes, colored boxes represents data from a distinct type of T cell, the CD8 T cells, the CD4 T cells, Tregs, NK cells, dendritic cells. Again, it's hard to wrap our brains around this. There's an awful lot of information there. I won't talk about spread. But this is what the similarity index looks like over here. And this is a beautifully designed panel. You see there's not a lot of overlap between the different fluorescent markers that we're using. We also have something called a spillover spreading matrix, where we're not looking at compensation, but we're looking at how the data gets stretched out between two markers that express very close to one another spectrally. This is becoming a common tool for us. So these are really just ways for us to go in and see how well our panel is working, whether we're going to be seeing good, well-delineated data or not. And here, Maria used, again, these probability state analysis tools to go in and attempt to visualize all the different subsets we're looking at in a single 2-parameter plot. We're able to identify 38 distinct subsets of immune cells using this 40-color panel. So each of these little numbered areas here represents a particular population. And then we can go back in and say, okay, are these T cells, are these B cells, are these monocytes? And looking at different, for example, in this case, different donors, we can look at the variability of this complex data from patient to patient to patient. Okay, so if you really want to show off, this is a 50-color panel. This is designed by Florian Mayer, who both Dr. Vivek and I know in Switzerland. Again, he's looking at a lot of different markers. The data is very, very complex over here, but a lot of information is being derived from this. And these are the different matrices, the similarity matrix, and also what we call the spreading matrix, to look at how well the panel is functioning. And again, he's relying on these data mining techniques, TISNI-VISNI, the probability state analysis software, heat maps, and other things to go in and identify different subsets. And if you were looking in a patient versus a healthy donor, for example, to be able to identify changes in these particular subsets. And as you can imagine, AI is now being applied to this, as it is being applied to everything in bioinformatics at this point, because these data sets are extremely large, and we have to rely on some of these tools to go in and see differences. And at my institute, we have a number of different instruments. So we've been testing them to see how well they work. This is my Aurora cytometer up here. The Sony system uses totally different technology. It uses diffraction gratings and a different type of detector, but the data is almost identical. So we have a lot of good tools out there for doing this type of high dimensional flow cytometry. So I don't have a lot of time left, but I want to talk about a couple other pieces of technology related to this that you should be aware of if you're doing flow cytometry in your lab. Don't worry too much about the 50-color data here. I have never done a 50-color experiment. I may never do one. I just want to show you how far you can take the technique. There are many, many things you can do with far fewer colors, but I just want you to be aware of the current limits, how far we're able to go using this technology. Huh? Yes. Oh, I mean, you know, we were, this sort of thing was applied actually at NIH where I'm at. They evaluated a lot of the early COVID vaccines, and they applied this extensively, this type of thing extensively to that, to understand problems like T-cell exhaustion. You know, why do some people get COVID severely and others are asymptomatic? The key is really in the structure of our immune system, and these tools have been applied widely to that. So the other pieces of technology I want you to be aware of, first of all, cell sorting. The instruments I have shown you so far take the cell, they analyze it, and then they throw the cell into the trash. It goes into a little catch basin and goes into a waste tank, and you throw it down the sink. However, you don't have to do that. You can analyze the cell and then use one of several mechanisms to capture that cell in a droplet and send it into a collection tube. So you can actually collect the cell population you're interested in. And if you're doing these 40 and 50 color panels, you can collect these different highly distinct subsets and do something with it. You can do proteomics, genomics. You can put it back in a dish and see what it grows into, what it differentiates into. You can inject it back into an animal and do something with it. So in cell sorting, we shoot the cells through a laser beam, just like we do on a conventional cytometer, but then we vibrate the nozzle that the cells pass through, and the stream breaks into liquid droplets. Now, because we know the time from when the cell initially hits the laser and breaks off in the droplet, we can charge the stream right before the droplet breaks up with a positive or negative charge, and then use electrostatic plates to divert those streams, those droplets, into collection tubes. It's a very powerful technique. It's actually been around as long as flow cytometry has been around. It's something my laboratory does a lot. We are a cell sorting facility. And I know when my investigators do some experiment by flow cytometry, in 6 months, they're going to want to separate out the cells. So this is what a cell sorter looks like on the inside. This is what we call a jet and air system, where the cells are shot out of a nozzle. The nozzle is here. The cells come in from the top. The laser beams are coming in from the left. The plates are down here. We vibrate the nozzle. The stream breaks up into droplets, and then they're diverted into collection tubes that are held down here in a rack. You guys have a Faxaria 3 downstairs, which is a very nice cell sorter. You've also got a Biorad S3 as well, which is a cell sorter too. So you have cell sorting capability in your department here. There are a couple different types of cell sorters out there. The jet and air shoots things out into the open air. The hybrid flow cell system actually analyzes the cell in a quartz cuvette. It gives better sensitivity, and then they're shot out into the open air. This is the system that you have downstairs. The Faxaria 3 is that system. And this is a Faxaria 3 right here, where you can see the cuvette where the cells are analyzed. The cells the stream passes down here. It's broken into droplets. These are the charge plates, and ultimately the cells are diverted into tubes down here. Now, fortunately, we have spectral sorters. Took a little while, but there are a number of systems out there. We have the Aurora CS in my laboratory, which is very similar to the analyzer. This is what it looks like in the inside. And again, what I love about cell sorters is you can open them up and see exactly what's going on. The charge plates are here. The stream will pass down here. Anything we don't want goes into a little waste trap in the center here. Everything we do want goes into collection tubes over here. It has the same optical bench as the analyzer I just showed you, which makes it very easy to move experiments to the sorter. And this is what the control panel looks like. And what you can see up here is the streams. These little white dots are those sort streams that have the cells that we're interested in, illuminated by a little infrared laser. And if you look very closely at this camera view here, you can see the cell streams being diverted into the collection tubes. We can instruct the instrument exactly what population we want based on those fluorescent tags that I told you about earlier, and collect the cells that you're interested in. This is what the stream looks like. It's frozen in time because we have a little strobe light that flashes at the same frequency as the nozzle is vibrating up here. So the cells are coming down here, they're separated into droplets, and we apply a positive or a negative charge just as the last droplet is about to break away. So once the drops break away, they have a positive or a negative charge. And there are a lot of little controls over here that we can adjust to operate the instrument. And we can get amazing purity. This is a 15-color panel looking at different types of T cells. This is what the unpurified cells look like up here. I've sorted my cells into CD4 positive, RA positive. These are naive, unstimulated T cells. And you can see we're getting 99% purity of the cell population we're interested in. And the same for the RO positive T memory cells. So it's really quite astonishing. We can get very, very pure cell populations. We can then sequence these. We can do next-gen sequencing. We could do proteomic analysis. We can put them back into an animal or put them onto a feeder layer and see what happens to the cells. We can add cytokines and see if they differentiate. When you open a textbook and look at all those hematopoietic lineages where the stem cells become pre-Ts and pre-Bs and then B cells and dendritic cells, that information was all collected the hard way by separating out these populations, putting them in a dish, seeing what happens to them, seeing if they differentiate. An awful lot of work, and cell sorting has been critical for that. You can also sort very rare populations. Here I'm using that same panel to collect a type of cell called a CD3 positive NK cell. There is such a thing. They're very, very rare. They're only 0.017% of all peripheral blood, but we're able to get 99% purity. Can't get a lot of them, but we're able to separate them out. Okay, so in the last 2 or 3 minutes, I just want to tell you what's next, because we're able to look at 50 markers at once, which is a lot. When I got into Flow, it was 2. And then it became, and we got all excited when it became 3 and 4. And now we're able to get these enormous amounts of information from our cells. The lasers are important. All the machines I've shown you, most of the more recent machines have blue, red, violet, green or yellow, and ultraviolet lasers that excite all these different fluorescent probes and let us go to 30 colors by conventional or 40 to 50 colors by spectral. So can we go higher than this? I think ultimately we're going to need to go higher than this as we discover more and more cell markers. So one thing my laboratory does is work on deep ultraviolet lasers. Currently, the shortest wavelength laser we use normally for cytometry is in the ultraviolet range, 355. We're getting to the point here where you can't see the laser anymore. So my laboratory works on lasers that are even shorter than this. And this is important because right now, although I haven't talked a lot about how dyes work, we can now design polymer dyes that will excite at any wavelength and emit at any wavelength. We can build dyes that will allow us to go beyond the 50 color limit. We're already using a series of dyes called the brilliant ultraviolet dyes that excite in the ultraviolet range and emit in the blue, green, yellow, orange, red. And these are very important for us, and there's some other, BioRad makes some as well. These are all synthetic dyes that have been specifically designed with these properties. And in fact, you can design dyes that will excite way down into the deep ultraviolet. And my lab has worked with most of these laser wavelengths, first of all, to see if they can work in flow cytometry at all, and then try to use them to excite novel fluorescent tags. And this is some of the research that's being done on this. This is a project from BD, which a company that makes a lot of flow cytometers. They're designing a deep ultraviolet dye series called the DUVs that excite around 300 nanometer. They're designed to do this in terms of their chemical structure, and then emit at different wavelengths in the green, yellow, orange, and red range. And we have a number of prototype lasers in my lab that excite way down in that range, in the deep ultraviolet down here, and in fact do a very good job of exciting these dyes. This is DUV 473, 527 prototype dye structures. They've been attached to antibodies, and these deep ultraviolet dyes do a very nice job of exciting these fluorochromes. And I want to go lower than that. We have specialized optics that are allowing us to go down to 266, 257, 244. These are lasers. You cannot see these with the naked eye, and you shouldn't get them in your eyes. They're not good. But we're now at the point where we're getting ready to add these additional laser wavelengths to the ones we already have and possibly go well above 50 colors. So we're not done yet. Even though 50 is pretty daunting, I think we can even go higher than that. We're not done yet with high dimensional. And there are a lot of factors that go into this. We have better instruments now. We're able to build those panels that work well, or all the dyes and the antibodies all work well together. We have better bioinformatical tools like probability state analysis algorithms and AI-assisted analysis. And there's a lot of good chemistry going on out there to design dyes that are compatible for flow cytometry. So I will stop there. Just to remind you, I work at the NIH in Bethesda, Maryland, in the National Cancer Institute. These are, there's Veena. Vivek was talking about Veena Kapoor. She retired about 4 years ago, and I swear the woman doesn't age. She looks exactly the same now as she looks in these pictures. But we are a service corps, and but we try to do some of our own internal R&D as well. We work very closely with our colleagues in other institutes at the NIH, with the companies like Cytec and Sony, and also with the laser manufacturers as well, to do some of our own internal research and development. So I know it's a lot to digest, but I can take any questions people have at this point about flow cytometry, and we can talk even a little bit about the machines you guys have here, because you have a very nice array of equipment of multi-laser high-dimensional cytometers on your campus here that are very good, very useful tools. So I'll stop there. Sure. Right. Yep, they're big. Mm-hmm. Yep. Let me show you. There's a typo here. This actually should be 213 nanometer. That laser is $300,000. It costs more than a cytometer costs. So we're not there yet. But UV, by adding more laser wavelengths, you get more colors, you get more power for analysis. So the ultraviolet has been a wavelength we've really, as Dr. Vivek says, it's very expensive. They're big. They're now smaller. They're still expensive. But lasers went from when I got started in flow, lasers were 2 meters long. They weighed 70 kilos. They needed cooling water. They needed high voltage electricity. Now you can hold a laser in the palm of your hand that's more powerful than the lasers I used when I started out in the field. And they're getting really tiny. I don't think they're getting much cheaper, unfortunately. But now we can produce any laser wavelength we want. That didn't used to be true. And it's, the earliest cytometers were developed back when lasers were still very rare. Lasers have been around for about 60 or 70 years. Okay. And I would say, practically speaking, only about 40 or 50. I mean, as one of my physicist friends said, if you are not in awe of the fact that lasers exist, you don't really understand them, because they don't just make light. They make a totally different kind of light than the light that's coming out here. And what has made cytometry possible is being able to concentrate a lot of energy on a very tiny area, because the light is coherent. So it's made flow possible, and it's no accident that lasers and cytometry kind of got developed around the same time. We used to use arc lamps and things like that, and it didn't work very well. So we are able to detect a few hundred molecules of some receptors using very bright fluorescent probes. The sensitivity is astonishing. And it keeps getting better and better and better, and a few thousand molecules routinely. So we can really get a lot of information out of it. But the deep ultraviolet stuff, you can't buy any of these yet, really. You can, but it's something we're experimenting with, because I think it's the future if we want to go beyond 50 colors, and some of us do. With genomics, we used to look at dozens of things, and now we look at thousands of things, and we might look at tens of thousands of things now. we're not, we can't at the time imagine we're really going to need to do it, but ultimately it does become something that we need to understand these systems. So we've talked today mainly about immunophenotyping of immune cells, which is the dominant application of flow cytometry. But people have used cytometers to look at plant protoplasts, parasites, bacteria, viruses, exosomes, microvesicles. We have a group in Ecuador we work with that looks at trypanosomes by flow. They have trypanosomes that express different fluorescent proteins. So they look at T. cruzi. Malaria is looked at extensively by cytometry. It's actually used diagnostically to look at the different life cycle stages of malaria parasites when they infect red blood cells and then they burst out the different, the merozoites and different stages. It's used clinically to look at leukemia, lymphoma, bloodborne cancers, and some other conditions as well. So it has many applications, and if you can get your cells into a single cell suspension and push them through a nozzle, protozoans, we'll dissociate solid tumors and look at the DNA content, protein content of these things. There are many applications that go beyond what we've talked about today. So, like microscopy, it's a tool in our toolkit. One of many tools, but an important one. Yeah. Apps. Mm-hmm. 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 Sure, absolutely. We didn't talk a lot about it today, but there's a large collection of physiological probes that can for example, there are probes that can intercalate into the plasma membrane and alter their fluorescent properties based on the electrical potential across the membrane. There's a lot of them, a number of them. So you can measure transmembrane potential in the plasma membrane, mitochondrial membrane potential. There are mitochondrial probes that will alter their fluorescent properties based on the charge differential in the inner leaflet of the mitochondria. There are fluorescent probes that are insensitive to that, but just label mitochondria. We have fluorescent protein systems that are organelle specific. They're used in microscopy, but they can also be used in flow as well. Fluorescent proteins, GFP, YFP, CFP, DS-Red, M-Cherry, there are hundreds of them now. And we have expression systems where you can express 6 or 7 fluorescent proteins simultaneously. Do your compensation or unmixing to separate the colors and look at complex gene expression events. We can look at cell death. We can look at cell proliferation. The list just goes on and on. We have many fluorescent tools, and this is an instrument designed to look at fluorescence. Yeah. Okay. Mm-hmm. 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 Sure. I'll answer and then Dr. Vivek can answer and give his opinion too. For something like that, the one drawback of cytometry is that you lose spatial relationships. You're looking at a suspension. You have to take the cells and break them up into a suspension. So if you're interested in that, and people are interested in that, that's why imaging exists. It might be difficult, but if you have a fluorescent marker for this, some sort of tag, some sort of fluorescent indicator, it can be done. You can look at plant cells. You have to separate them into a single cell suspension, which can be difficult, but it is done. Plant cell cycle, looking at ploidy using DNA binding dyes, is very commonly done in plants. Looking at triploidy, it has commercial implications. We're working with groups that do that. GFP has been expressed in plants, and you can look at it by cytometry. So if you have an antibody or a marker or something that is going to provide fluorescent information, it could possibly be done. But you do lose the spatial information. And that's where microscopy comes in. Yeah.
No, aneuploidy you can, because you will start seeing shoulders. Yeah, then you can. If it's tetraploidy, then that's easily measurable. But then if all of them are tetraploid, then you lose heterogeneity information. And if the best way is that if you have some markers, and so even with the stem cell community, we kind of struggled with this a lot, especially outside the hematopoietic system. The hematopoietic system, people have designed very good markers. So if you have a marker that tells you this is a cell that does this as a function, then that heterogeneity becomes easier to explain. But if you, and then you can of course detect it. If you don't have markers, then it becomes more difficult.
This is from there we go. This is actually data from a colleague of Dr. Vivek's of mine. This is from Ed Agarwal, who actually, I guess he gave me the slide. This isn't his data, but he does cell cycle. So you can look at tetraploidy, triploidy. So these are plant cells that have been reduced to a single cell suspension, labeled with a DNA binding dye, like propidium iodide, and then analyzed by flow cytometry. So the peaks that you're getting here are the G0, G1 DNA content. But by using a set of internal standards, you can determine whether it's diploidy or triploidy or tetraploidy. This is probably the big application for plants. But there's really, again, if you have a fluorescent marker of some sort, there's no reason why you couldn't see it based on flow cytometry, using flow cytometry. This is T. Cruzy, Jaime Costales Cordero, who's the head of the Latin American Health Institute in Quito, Ecuador. That's a cytometer we provided him with, and he's able to look at T. Cruzy subsets. He actually has ones that express GFP. So he does drug testing, toxicological testing, looking at viability in T. Cruzy, and they're very easy to distinguish, actually. Malaria is the techniques for identifying the trophozoites and schizonts in malaria are very well worked out. There's a large body of literature on this using DNA and RNA specific dyes. And this is even used clinically actually now. So there are many different, all you have to do is get the cells into a single cell suspension somehow. It's not replacing microscopy, but it's an adjunct technique.